is in terms of the syllabus. There have been some slight changes there. Instead of Dr. Estep from OHSU, who's going to be giving a guest lecture, it's going to be Dr. Ruedas from our department. He's going to be talking about virus ecology, and particularly virus vector ecology, which will tie in nicely to some of today's talk as well. I have midterm scantrons for people who haven't had a chance to pick them up. You can get them then after class today. So didn't have clicker questions last time, but today we do. So yay, exactly. So where does poliovirus usually replicate? Neurons, gastrointestinal tract, upper respiratory tract, lower respiratory tract, or the oceans? What does entropy have to do with poliovirus? Okay, just checking. Yeah, exactly. That's good. It's all good. So, 10. Okay, yes, polio is a GI disease. Replication in neurons is a problem in terms of the symptoms that you usually see in terms of polio. But it's really quite rare. It's not replicating in neurons really much at all. Definitely a GI tract, which is one of the reasons why the inactivated vaccine is not as good in terms of getting herd immunity as the attenuated, because you actually end up getting people who are shedding the virus here. Upper respiratory, lower respiratory tracts, yes, there are picoRNA viruses that replicate there, particularly what kinds of picoRNA viruses? The rhinoviruses, exactly. And then the oceans, there are lots of these picoRNA viruses in the oceans. We don't know what they are doing there, but clearly polio is, as far as we know anyway, not a big deal in terms of the oceans. So next question, how many open reading frames are there in rhinovirus genomes? 1, 3, 5, 16, or 23? Good, we all agree. What is that one open reading frame? It's the polyprotein. So one open reading frame, that's it. That's true for at least these viruses. We'll talk about some other single-stranded RNA virus genomes that have multiple open reading frames that are used. But at least for all of the picoRNA viruses, and of course rhinoviruses are part of that, it is 1. So let's hide these guys, and we'll take our last one for today. The reason that the Sabin polio vaccine does not usually cause disease is a change at the 3' end of the genome, a change at the 5' end of the genome, a large deletion in the middle of the genome. It does not bind to VPG. It encodes a defective 3C. Ten, 5, 4, 3, 2, 1. So a little bit more of a difference of opinion on this one. First question, Sabin, inactivated or attenuated? Attenuated, right. That means it's replicating, right? So that's one thing. That, okay, doesn't rule out any of the rest of these. And doesn't usually cause disease. That's because occasionally you get the reversions. Very, very rarely, but occasionally you can get those reversions. So what's the change? And the iris, which is what? Well, so it's the internal ribosome entry site. And where does that have to be in terms of where your ribosome is going to be? Has to be at the 5' end. If it's at the 3' end, the ribosome gets there, and what does it do? It doesn't translate anything. So has to be at the 5' end, and so these other things are not true. So yes, it's the change at the 5' end of the genome. Those of you who have to leave, that was the last clicker question for today. Adios. Have a nice weekend. And good luck on whatever other exams you need to be dealing with right now as well. What other, is there is it cell biology today? Yep. Okay, good luck. Anyone else needs to take off? We're good now? Now for our next clicker question. No, I'm not that mean. So today we're going to talk about the Flaviviruses, and basically, as far as I'm concerned, Flaviviruses are really picoRNA viruses with a membrane. That's about it in terms of the differences between these kinds of viruses. The other, of course, probably more interesting aspect, certainly from an epidemiological point of view, is that there are a number of emerging diseases, etc., which are these Flaviviruses. West Nile, Hepatitis C, Dengue, and yellow fever are sort of the major ones in terms of those particular diseases. The key concepts that we have today really have to do with that difference between the picoRNA viruses and these Flaviviruses. These guys have membranes, so entry is clearly going to be different than the picoRNA viruses. They've got an envelope. The other ones don't have envelopes. What's really important and really different here relative to, again, the rest of the picoRNA viruses is that cellular proteases are also a very important component of how these viruses replicate. And, of course, membranes have to do with that as well. Classic, again, breakdown here. Here's a cute little yellow fever virion over here. So we'll talk a little bit about origin and diseases. We could spend a really long time talking about diseases, and I'll try and get through this relatively quickly. Structure, binding and entry, again, because these are enveloped, this is a little bit different. Replication and translation are basically identical relative to the picoRNA viruses. And of course, assembly is going to be different because these are enveloped viruses in terms of how the virions are. And again, this, you can see the envelope around the outside here. It's not a nice icosahedrally symmetrical form here. Why do we care so much about flaviviruses? Basically because of all of these diseases, human diseases in particular, although there are a number of other animal diseases that are associated with these. And the ones we're going to talk about today are yellow fever, dengue, West Nile, and hepatitis C. So of those, almost all of these are so-called vector-borne diseases, and in many cases also arboviruses. And this will be the viruses that are being transferred by insects. So it's almost all about our friend here, the mosquito, which takes blood from one animal and transfers it to another one. Again, usually in these cases, they're going to be humans. The big exception to this is hepatitis C, that seems to be basically just a blood-blood connection as opposed to going through these mosquitoes. And that means that controlling the spread of a lot of these diseases, in particular yellow fever, dengue, and West Nile, it's much more about controlling the spread of the mosquito than it is controlling the spread of the virus per se. Now, that being said, it's a lot easier said than done to control the spread of mosquitoes. And hepatitis C is a really fascinating virus. It's really quite recent, and we'll spend some time talking about it. It's also been quite a bit in the news recently in terms of some antivirals. So start out talking about yellow fever. Main reason for this is it's the one we've known the most about. And in fact, it was one of the very first diseases of humans to be shown to be caused by viruses. So it was way back in the late 18th and early 19th century where it was introduced into the New World, causes organ failure and acute hepatitis in a very few cases. It's actually relatively few that you have that. There's a vaccine strain, the 17D strain, which we're not working with in my lab yet, but potentially will be in the relatively near future. It's extremely similar, again, to the wild-type virus. It was, again, one of these viruses that was an attenuated virus made by doing multiple passages through both cell cultures and animals. Only 32 amino acid differences in the whole genome, mostly in the envelope protein. Again, that's what's on the outside on the surface. Much more recently, there was an infectious clone that was developed, and I wanted to spend a little bit more time talking about infectious clones because I don't think we've covered that yet. Basically, it's that you can make a DNA construct and then either in vitro or in vivo transcribe that DNA into an RNA, and that RNA is then infectious. So we talked a little bit about this for polio. It turns out it's actually harder to do this for polio than it is for something like yellow fever virus. So this is, in fact, a quite straightforward process. This vaccine is great, though. Provides great immunity and is extremely safe. I don't really know. I think there may be some very, very small amounts, maybe 1 in a million kind of cases of this being a problem. There were some interesting issues with the discovery of yellow fever being a viral disease. The main problem was it wasn't directly contagious. So if somebody who had yellow fever couldn't actually transfer the disease, obviously, to someone else in the absence of mosquitoes. And there's all kinds of interesting studies led by Walter Reed and a few other ones where they would put people in rooms with mosquitoes and rooms without mosquitoes. Again, things that no way would ever get past an IRB in terms of doing these kinds of experiments. But, you know, presence of mosquitoes, you had transmission of disease. Absence of mosquitoes, you didn't. Later it was shown that blood contained it. And again, these were volunteers. How they were volunteered was an interesting question. And people thought for a long time it was just replicating in humans, which makes studying the virus, of course, really hard if you can only use human subjects in terms of the process. Fortunately, mice, about 30 years later, was shown to be a nice host. And there's been huge amounts of work that's been done on this. Fortunately, yellow fever is not endemic in the US, but there are lots of places that people from the US go in which yellow fever is an endemic, and vaccines have not made it there, unfortunately. It turns out that one of the reasons that vaccines haven't made it there is that this vaccine is really pretty unstable. As soon as you reconstitute it, you have to, so it's made lyophilized, so freeze-dried. As soon as you add water to it, you have to use it within a couple of hours. So this is a target for some of the zombie virus technology that we're developing in my lab. So that's why we may, in fact, be using this particular strain. So, but generally, yellow fever is not much of a problem unless you're going somewhere and the major people in the US are getting it are military at this point. Dengue, on the other hand, is a much more significant problem. Dengue fever, or dengue hemorrhagic fever, which is one of the more major problems that you get. There are hundreds of millions of infections in any given year of dengue fever. So this is a really, really big deal, and we'll compare this to what's going on in terms of hepatitis a little bit later on. Of those, about 500,000 get the dengue hemorrhagic fever, which is a very dangerous disease. 20,000 deaths in any given year, and most of these are unfortunately children. Due to transmission, fortunately, actually for us, is, particularly being in the US, is that this is almost all through only 2 species of mosquito which can transmit the disease. And so we'll see in just a second that it's control of these mosquitoes which is probably going to be the way that we can deal best with this disease in the absence of having a good vaccine. As I mentioned before, dengue hemorrhagic fever happens much less frequently than normal dengue infections. Usually has to do with multiple infections of multiple of the 4 different kinds of dengue fever happening in the same place at the same time. Dengue shock syndrome is an even more extreme case, and that's what's usually leading to a lot of deaths. And these are numbers, by the way, from the WHO and CDC. We'll look at some of those data in just a second here. If you look at dengue fever, it's a really interesting example of an emerging disease. Prior to 1960, it was basically present in about 4 countries in the world, depending on how you count all these countries in the Caribbean here. After 1960, it spread basically to almost all of the tropical countries in the world, and some even, really quite subtropical in terms of the spread of the disease. And if you now look at where the mosquitoes are relative to this, they're basically overlapping with each other. So Aedes aegypti is one of those 2 species of mosquito. All the yellows here are Aedes aegypti, and then red is Aedes aegypti and epidemic activity. That was just when this particular slide was made. This particular one was in 2005. Basically, everywhere where you find Aedes aegypti, you will also find dengue fever. And you'll notice, for those of us who are US-centric, that there are a few places where we have Aedes aegypti in the US, but it's pretty few and far between. So I wanted to take a quick look here. One of the things that I think is excuse me, a really wonderful resource are some of the maps which are put out by CDC, by the US Geological Survey, and this gives you really up-to-date information on where activity is happening. You'll notice a few cases here, but almost all of these are imported cases. But otherwise, pretty much throughout the whole tropics, you've got these infections going on. Yeah?
So the dots, are those outbreaks, and is the diameter basically how big? Yeah.
Basically, yes. So if we go back and look at, I don't know if the list view Here's our settings. Again, this is, I don't know, fun to play around with, might not be the right term, but there's a huge amount of information here, and it's really quite well taken care of. That we know where these things are certainly doesn't mean we know how to treat the disease, but it's certainly something we're looking at. Yeah?
Why can only 2 species of mosquitoes carry it? Can it kill the other ones?
So it appears that the reason for that is the replication cycle, so that these, all of these viruses which are dependent on the vectors undergo replication in the mosquito. And so there are presumably some cellular differences between the different species then that allow them to replicate or not. Is there another question over here? Yeah.
I was just going to ask why there's an outbreak in the middle of Siberia.
So unfortunately, these dots represent countries.
Okay.
And so that's cases in Russia,
Okay.
as opposed to exactly where they are. And so this one, I think, is the Maldives, islands off the coast. So the dots themselves are going to be, here's, case in Northwest Territories in Australia. It's almost definitely up towards Darwin up here at the top where these are actually happening. So you can get higher resolution versions of this, but that's, again, the main thing here is just to show you where you can get information on this. So dengue, as far as I'm concerned, is probably one, if not the most important global viral disease at this point. So there's I think a lot of research going on in terms of understanding dengue, but still a long way to go. On the other hand, we hear a lot more about things like West Nile, and as we'll talk about next, hepatitis C. West Nile is an even more amazing example of an emerging disease, particularly in the U.S. Was known in Uganda in the 1930s and probably has been endemic in Central Africa for decades, if not longer than that. First case in the U.S. was in 1999 in New York City. 9 years later, there are cases all the way across the country. And you can follow the process, basically going from New York, and Oregon, curiously enough, was one of the very last places in the U.S. to have cases of West Nile. West Nile disease is generally not a big problem unless you're a bird. And it's really devastated some of the bird populations, particularly in the upper Midwest. And any of you who have been here, and in fact, a lot of these places, you know that this is absolutely loaded with mosquitoes. And the correlation between the amounts of mosquitoes and the numbers of cases are really very, very high. Any idea why you'd be having this clump down here? Yeah, a lot of this is irrigation. And so irrigation, what do? Open water, mosquitoes. Again here, Central Valley of California. Irrigation, open water, places where mosquitoes are replicating. We're in, fortunately, pretty good shape up here. Not too many mosquitoes, but again, population density through here in Idaho. So that's a good example. Here's another. I just wanted to throw up the link. You have it in your PowerPoints. You can see where West Nile virus is in the U.S. Currently. And this is updated on, I think, about a weekly basis. So they're doing a really good job of surveillance and keeping track of that. So the last one of these Flavivirus diseases I wanted to talk about is hepatitis C. And this is different than all the other ones because it is apparently just direct blood transfer, not requiring these mosquitoes. And the way to be infected with hepatitis C is large amounts, usually, of infected blood. And so most people, unfortunately including me, which gives you a little bit of my age, have been exposed to hepatitis C. And those are people who are much more likely to have hepatitis C. There were, in 2007, about 17,000 known new cases. There's a relatively low fatality rate, and we'll get back to this a little bit later on. Therapy here is actually from your textbook. It's somewhat outdated now. Turns out there are some really very good therapies. relative to hepatitis C. But in most cases, hepatitis C doesn't seem to cause much in the way of disease. It's a latent process. It's very slow. Liver degradation, again, a hepatitis process, is certainly a very large contributor to liver cancer, liver transplants, etc. But the fatality rate, particularly given the predicted hundreds of millions of people who are infected, is really quite low. So there's been, I don't know, hype maybe not be quite the right word. There's a really nice link here at the CDC talking about hepatitis C virus. I personally think that the health risk for hepatitis C is really not that high, and we only discovered it, in fact, really quite recently. We've got cDNA clones, we can study it quite well. But that being said, as again some of you may have seen in the news, there's a new anti-hepatitis C drug called Flavi. It's an inhibitor of NS5B that we'll get to in just a second, approved by the FDA in the end of last year. Since then, almost $2.3 billion worth of this drug have been sold, which is really pretty insane in terms of the amount. In fact, it's the fastest selling drug ever at this point. There are also competing drugs that were also just approved in the last couple of years. Incivek, we'll go and take a quick look at this website because I just think it's just too, well, you'll see what I'm thinking about in just a second. So the graphics here are just fabulous. So this is how, so it's, yeah, you're fighting off the disease, et cetera, curing hepatitis C. Hepatitis C infections are probably infecting maybe a couple of thousand people in the U.S. now in every couple of years or so, and it's usually an underlying disease which is causing problems. Now, you remember what I said about dengue? Tens of thousands of people, 500,000 people a year coming down with these symptoms. But this is a developing world thing, or a developed world thing, I should say. Hepatitis, developed world, people who are living longer have issues with this. The big issue with Flavi, excuse me, let's back up here. Any idea why it's this $2.3 billion? It's very expensive. How much does that one pill cost? You heard the news? It's $1,000 a pill. We can talk about that, yes.
Do you know the process of making it? Is it because it's an effort to make, or just because they're greedy?
So the question is, why is it so expensive? And the answer is, at least the company, Gilead Biosciences, can't give you a good answer on that. It's a relatively small molecule. Making it, I don't know, but I'm guessing it's really pretty cheap. It's the process of development in that. And again, this is a developed world disease. You couldn't be selling $1,000 a pill in the developing world. So there's some interesting ethical issues here too, yeah.
Do you have to take it all the time or just once?
The current regime is, I think, 84 days, once a day.
Is it one pill a day? Or is it?
One pill a day, yes.
Oh, okay.
So, yeah, you can do the math on that one. So, yeah, again, we can talk more about that offline if people are interested. So now the interesting stuff. Now, forget the disease. Let's talk about how these things are put together, although it is in the understanding of the molecular biology of these viruses is how these drugs have been developed. And so that Flavi, as I mentioned, is an NS5B inhibitor, very specific inhibitor to one particular protein in the hepatitis C genome. And the other one, the boxing glove, is a protease inhibitor. So structures of these flaviviruses, they're enveloped, but curiously enough, even though they're enveloped and they have this, it's again, envelope's got a membrane on the outside, it looks a lot like a naked virus in terms of the structure. Basically, that envelope is packed chock full of these envelope proteins, which are present on the outside. And these are those capsid proteins here on the outside, I should say the envelope proteins, excuse me, which are arranged as these dimers in a pretty classic T=3 icosahedral form. So usually when you think about an enveloped virus, you think about lipids and being really mobile, but in this case, they're pretty well locked down. On the inside is a nucleocapsid, and that capsid is then also enclosing the DNA. So you've got the envelope proteins, and then on the inside, a nucleocapsid protein as well. If you just slice one of these things open, my apologies for the German at the top here. It's the nicest image I could find. We've got our envelope proteins on the outside here, a lipid bilayer, and then our nucleocapsid protein followed by the viral RNA on the inside. This is all from cryo-electron microscopy, and the fact that you can see density for the RNA on the inside, if you think about cryo-electron microscopy, it's averaging many, many, many particles. So if the structure of the RNA here were different from particle to particle, you would never see any density here. And the fact that you do see density, again, means that these are really, really structured in terms of the envelope proteins, how they're all aligned with each other, and then also the nucleocapsid and the viral RNA, which is present on the inside. Let's start out looking at the E protein, which is the major structural protein. On the outside, the envelope protein. This is really different than the envelope proteins that we've talked about so far, things like hemagglutinin, or we looked at entry before. These guys sit down flat on the membrane. So this is basically looking down at the membrane, the envelope on the outside of the cell. These two dimers sit right next to each other. They have fusion peptides down here at the end of each of the molecules. And again, this is a dimeric structure. This was all worked on by Steve Harrison's lab. We had the links when we talked about the entry lectures that we had right towards the beginning. Lots of information on exactly how they've solved these structures. So let's look at the entry process here. The process is receptor-mediated endocytosis. Unfortunately, it doesn't have any of these on here, but the process is very much like what happens in a case of hepatitis B. You have binding to cellular receptors. Once you have that binding, you come into the endosome. These guys get released from the endosomes, and then that RNA is in fact replicated not in the nucleus here, but in the cytoplasm. So it's a sort of this process here. One of the big questions is what's the receptor, or what are the receptors, I should say, for most of these flaviviruses. It's really not clear what most of these receptor proteins are. So that's a big area of research. One of the things, however, we do know, and this is particularly true for dengue, is there's something called antibody-dependent enhancement, which is really bizarre. And that is, if you have antibodies to a virus, it actually leads to more uptake by the cells of the virus than less, which again seems really kind of bizarre, and evolutionarily how that happened is not completely clear. But again, what happens is you have antibodies. If you have circulating antibodies to a particular virus, that then will get taken up more rapidly inside your cells than otherwise, and this may be why people get dengue hemorrhagic fever. And as I mentioned before, the places where you usually get these big outbreaks of dengue hemorrhagic fever is you have the multiple types. I mentioned there are 4 different types of dengue. If you have infection by one of the types, then you'll develop an antibody response, and then you get infected by a different type. That leads to this antibody-dependent enhancement, and probably why you're getting a feverish response, so the hemorrhagic fever, as opposed to the infection by a single dengue virus, which usually doesn't lead to a massive amount. And this is also probably why, even though we do have in the U.S. Aedes aegypti and the other one, which our name I've forgotten now, the other Aedes species, which carries dengue, the amount of infection is really pretty low, and it's usually just one or maybe at most two of the different strains. So at that point we have dengue fever, but very little of this dengue hemorrhagic fever because you have multiple ones that are happening at the same time. So probably involved in this entry process leading to much more severe disease. So I mentioned the E proteins in a little bit more previously. These are present as dimers on the surface of the virion. But dimers are not really what we've mostly talked about in terms of entry into the cell. And in fact, I showed you the video when we talked about entry of dengue entering the cell. The really, I thought, great animation of the fusion and then the release of the RNAs with the virus proteins that are attached to them. That's a trimeric process. So how do you go from dimer to trimer? Now, it turns out that this is just what's happening in the endosome. Endosome, what happens? The pH is lowered. Once you have the pH being lowered, you have a conformational change. But this particular conformational change is way more than you have with something like influenza virus HA that will pop a fusion peptide to the top of the protein. In this case, it goes from dimers sitting down on the surface to trimers sticking way out of the surface. And if we have a chance, we may take a look at that video again to take a look at that process. And basically, this is just another cartoon looking at the same issue. Here's the dimer sitting down on the surface of one of these flaviviruses. Here, looking at it side on. Here, looking at it top on. This is in the normal neutral pH state, which you're going to have on the outside of the cell. However, in a low pH, which you're going to be getting in the endosome, this is the kind of structure which you get, which are 3 of these now, with the fusion peptide down here at the one end, actually looking a lot like influenza HA or the HIV proteins that are involved. And then you have this rearrangement process that takes place and eventual membrane fusion. And so we'll take another look at this movie again because it's just so cool. But again, the important thing here is to look at that change between.
It affects millions of people worldwide and is
that we don't need. But look at the dimer to trimer transition that takes place on the entry process as these virions come into the endosome and that process takes place. So here the virus is in the endosome being transported inside the cell. Here we're dimeric, we're flat on the outside. Here's all the hydrogen ions coming in, and you see they completely reform into these trimeric structures. So now the trimer, which now has this fusion peptide at the end, these trimeric structures now can associate with the membrane of the endosome and then have this amazing secondary conformational change which pulls the two together. So the primary conformational change is going from the dimers and trimers and sticking up these fusion peptides to the top there. The second change is now pulling those two membranes together, getting membrane fusion to take place. And then again, once you've opened this up, now here's your RNA together with all of its RNA-bound proteins. It's coming outside of the cell. Yeah?
So the dimers that turn into trimers work in pairs?
Yeah. So you go basically from 3 pairs of 2 to 2 pairs of 3. Okay, so what's released inside? This should look really, really familiar. 1 polyprotein, 1 big open reading frame. There are a couple of differences, however, and probably the most important one is down here at the 5' end of the genome. It has a cap structure, so it's not a protein, which is bound here. It's a cap structure, which it turns out is slightly different than a lot of the caps that you usually have on messenger RNAs. Another difference is down here at the 3' end, there's no poly A tail. So it's just a cap at one end, no tail. You have to somehow protect this end. That's one of the things that poly A tails do. The way that that works is also the way that it happens in probably most rhinoviruses, is that you have a secondary structure at this end. And it turns out that this sequence right here is complementary to this sequence over here. So complementarity between the 5' end and the 3' end, which will loop up your viral genome. And again, this should sound really familiar because it's a lot like what you get with cellular messenger RNAs. And so when one ribosome gets to the end of this genome, it can hop back right on at the opposite end. Because we have a cap structure, we can use all of the normal cellular cap binding enzymes to get normal translation. Some of these have iris sequences as well, but for the most part, since they've got caps, you can have the normal process for how these are getting picked up. The structure of the genome is very similar, again, to what we saw in the picoRNA viruses. We've got our structural proteins, or I should say the structural preproteins, because it's all one big protein, at the 5' end of the genome, and then all of our non-structural proteins down here at the 3' end of the genome. So polyprotein, what do you need to do? You need proteases. You need to cut them apart. The viral proteases, NS3 and 2B, I wouldn't bother remembering all these names of these things. It gets worse and worse as time goes by. But the big difference here is that there are cellular proteases that are also important for making these final genomes. Which are the proteins I think you should be thinking about? There's the envelope protein, which we just talked a lot about, dimers going to trimers, sitting down on the membrane, popping up with fusion peptides. This M protein that we'll talk about in just a second. And this last one down here, the RNA-dependent RNA polymerase. But not only RNA-dependent RNA polymerase, it's also a methyltransferase. And that methyltransferase is how you're making the cap structure. So as I mentioned before, once you get entry, you're released into the cytoplasm. There's no cellular mechanism for making caps in the cytoplasm unless you bring it with you in your virus genome. And so the capping that takes place on all of these flavivirus genomes is due to viral proteins. It's not due to cellular proteins. Where does replication take place? Happens on the endoplasmic reticulum, and the main reason for that is, of course, we have our envelope protein, and so envelope proteins have to be associated with the membrane, and the membrane that ends up around these flaviviruses is the endoplasmic reticulum membrane. And what's interesting is that instead of being produced at the outside of the cell, it's being produced here in the Golgi and basically being secreted as if it were a secretory vesicle. And it's being released in that particular process. Again, we already talked about how you get binding. The genome is released. Again, it's capped, so you can just have cellular translation. They'll make a polyprotein, and then that polyprotein will get targeted to the endoplasmic reticulum. What happens in the endoplasmic reticulum is that you have production of all of the envelope proteins at that place. Yeah?
The pH change as it passes through the Golgi doesn't cause it to bind with the membrane again?
So during the trans-Golgi process, the pH changes that happen in the Golgi are generally considerably less than what you have in the endosomes. And so that doesn't seem to be an issue. Now, what is happening in that process, and in fact going through the Golgi has a number of cellular proteases in it, which it turns out are really important for this process right here. And so if you think about the, now as you're going through the Golgi, and this, so you have to just back up a little bit. So when you are making the polyprotein, you have all of your structural proteins that are being made here at one end, and then your non-structural proteins down here, which are further along towards the C-terminus of your polyprotein. So this inserts into the lumen of the ER, and remember the lumen of the ER, that's what's going to be on the outside of your virion. So when that's getting transported out to the Golgi, that's where you have cleavage, and the most specific one here is the furin protease. So that will cleave in the Golgi, and it turns out that that cleavage right here releases the E protein so that it can fold up into the form which now can undergo the conformational change when it comes back in. And so it's that proteolysis that's taking place during that transport to the Golgi is also called the maturation of the virion, which allows it to make that contact later. Yeah?
So the import in VR, is that the translational?
Yeah, it's very just like you would think about in other classes that may be having midterms. So yes, very much so. So that process is happening, and you also have the activity of the signal peptidase, which is exactly what you'd have with other membrane proteins that you're producing in the ER. So great review for cell biology. It's a good thing you stayed. So you've got signal peptidase, and then this furin peptidase that happens much later on in the process, and it's this M protein which is basically blocking that conformational change that you would be having as you're going through that Golgi process. So that's the structural parts. The non-structural proteins, basically the important ones are NS3 and NS5. Why are they so important? Well, it turns out this is the target of that $1,000 a pill drug, and this is the one with the competition here. So NS3 is basically the equivalent of the 3C protease that you have in all the picoRNA viruses. It's a protease, but it also has helicase activity, nucleoside triphosphatase activity. NS4A and B is not that critical. NS5, as I mentioned before, it's important as a methyltransferase, but it's also the RNA-dependent RNA polymerase, and of course, the target for a number of these drugs. There are other non-structural proteins which turn out to be really critical for virus function, but they're not in red, so don't try and remember all of them for the midterm. No, I didn't say that. No, but so there are these other proteins. They are very important for getting virus replication to take place, but the really critical ones are NS3 and NS5. So we basically covered this answering your question that we have assembly at the ER. You end up with the genome being packaged on the outside of the ER, so you have all of these transmembrane proteins that are being produced again through the translation at the ER, which is then inserting all of these proteins there. The C protein, which, sorry, I should have included that here. The C protein is now in the cytoplasm, because that's the very first thing which is being produced in your polyprotein. This C protein is the nucleocapsid, so this is going to bind to your RNA. So your RNA is all out here in the cytoplasm. All of your membrane proteins are out here in the lumen of the ER, which eventually, again, is going to be where they get transported to the outside of the cell. Budding from the ER is really standard budding. In the Golgi, that's where you have the furin peptidase, which cleaves the PRM protein, giving you your final structure. And then exocytosis, again, very much like a secretory vesicle, which you would have in any particular case here. So that's all of the molecular biology of these flaviviruses. Since we have another couple of minutes, I did want to show one more video. And so in HCV, no idea what in HCV and who's actually promoting this, but oh, sorry about that. Let's shift this over to the other screen. So here's the liver, again, potentially undergoing disease. Here's our, I don't know, is this dimeric or trimeric? Hard to tell in this particular image here. But that's the virion structure. Here it is going to try and find a cell. Now, this is obviously the really horrible current therapeutics. We're going to hear about all the really good inciting ones just now, so we keep listening to the rest of the video. So again, it's all about protease and the polymerase inhibitors. So here we have our hepatitis C virus in blood. Of course, it's not infecting the red blood cells, but that's a different story. Here we have interaction with, again, as far as we know, unknown receptors. And again, people are really interested in this, but we don't really have a good handle on how this binding is taking place. Again, this receptor, whatever it is, leads to receptor-mediated endocytosis. We have the virion getting inside the cell. Once it's in the endosome, again, we have lowering of the pH, release of the capsid here, all falls apart. Now, how nice and wonderful. Here's our virus genome, which of course should really be circular. This is supposed to be a ribosome. I'm not quite sure how that works, but again, there's ribosomal processes. Now, these are the non-structural proteins, which have all been made at the membrane. And at this point, this is before we have the proteolysis effect that's taking place. And again, NS3 and NS5, in this case 5B. NS3 is a protease, NS5B is your replicase protein. So these are all now supposed to be the fusion proteins, which have not been cleaved yet. That flash was your protease, which now allows these enzymes to replicate this little worm, which is your viral genome, making many more copies of it. The replication process, by the way, is almost identical to the picoRNA viruses. Now, here are their investigational agents, i.e., I'm almost certain this is a company which has put together this video. It's way too flashy to not be that process. So this is a potential protease inhibitor. And a quick look here. Again, it looks a lot like a potential protein. You've got these peptide bonds in there. And so basically, it binds to the protease as if it were a peptide, but blocks the activity of the protease. So if you block the activity of the protease, of course, you're not going to be breaking down these individual non-structural proteins. If you don't have separate peptides for these things, they're no longer going to be active. This is, so basically, that's the first process. The second one is addressing the NS5B, the replicase protein, again, binding to the structure of this protein in order to block the RNA-dependent RNA polymerase activity. In the video, and I'll give you the link here posted on D2L, they also talk about the palm, finger, and hand structures because, again, just like the case that we have with the picoRNA replicases, they're DNA-like polymerases, and this particular small molecule potential drug is blocking that activity that you would have in one of these normal DNA polymerases. And the trick for a lot of these kinds of drugs, particularly for these polymerase inhibitors, is they're DNA-like polymerases. Well, you need DNA polymerase to be able to be replicating. So you have to have something which is really specific for this viral polymerase relative to the cellular polymerases. And so that's probably the real trick in terms of getting these things to actually work. So again, the boxing glove version is the protease inhibitor, and the $1,000 a pill is the one which is blocking the RNA-dependent RNA polymerase. Have a good weekend. I'll see you on Monday.